Okay, yes, we have a lot of ground to cover. This is a complicated topic. All of them are, but I've been wrestling with this one, and I want to leave some time at the end for questions, so we'll do our best to get through. Welcome, I'm Logan Ward. I'm a contributing editor for the magazine, and I've had the privilege to work on the Popular Mechanics Breakthrough Awards every year for 8 years since the inception. And I was thinking about it with the memory of the Olympics from the summer in mind, and I was like, well, this is sort of a, an Olympics of human progress. We stick mostly to this country, but in terms of the teams and the variety of innovation, it's incredible. So every year to be able to come here, meet them, and hear them speak is a real treat. This particular team of winners really struck me on a variety of levels, so I was thrilled to be able to moderate this panel. These are 5 incredible people. What they've done really pushes science fiction into the realm of reality. Simply put, they were the first to demonstrate that a human subject, Tim Hemmis, could move a robotic arm using only his thoughts. And we're going to try to understand how that came to be over the course of this panel. Now, instead of introducing each of these gentlemen and throwing out all their credentials and all the abbreviations after their names, I thought that I would tell this story as a story in the sort of chronology. So I will introduce them each as I get to their role in this story, and then let them speak about how they got involved, and we'll take it from there. Okay, so in the middle here, he is a professor of neurobiology at the University of Pittsburgh. He's been decoding brain activity related to conscious thought for decades. In fact, when I was boning up on his career, I learned that his 1984 PhD thesis was titled Activity in the Deep Cerebellar Nuclei During Normal and Perturbed Locomotion, which I guess means
endless amounts of the future, still push forward, but it worked. All this hard work from Andy, and I mean, it works.
Yeah, and just to be clear, I mean, the arm was sitting somewhere else. I mean, it was not attached to your body. It wasn't functional.
No, it was
per
sitting
se.
beside me. Yeah.
And you told me, Tim, that you knew from the very start that this particular study was not going to help you in any way, in any concrete way. It was just going to be a way to further the science and to get in with this team. And it seemed like all along you've had this sort of long-term thinking. Along those lines, I want to get back to Mike Boninger. So this is pioneering work, but it's still very rudimentary. It only lasted 30 days or 28 days for Tim. How do you see this technology evolving into something that helps people directly, and how long might that take?
The how long question.
Mm-hmm. We have to ask it.
So I think that this technology, the first thing that we need to do is work on getting more than 28 days.
Yeah.
Because what kills me is that we really made a lot of progress. Wei led this team, and under his leadership, after about 14 days in is when we really started to take off. And so if we had a year, what would we learn? How much further could we go with this? I think when you get stable control, for this technology, it's not hard to get that amount of signal out of the body. So obviously, what you'd hope is that one day you'd have a device that was fully implanted. I think that you could get to a device that helped in function within a couple of years, to get to a device where I can look at a patient and say, here's one of the options. This is a clinically available tool. I think is in more of the 5 to 10 year time frame, but that's already something, and we're building off of what we have, which is something real that happened, and so we want to get it implantable. We want to get it 5 to 10 years, and the other thing we really want to do, just to go back to something that Tim said, is we want to make sure that you can feel it. Yeah. In the absence of the sensation, we're not going to, it's going to have a limited amount of function, a beautiful thing, right, for someone who can't move, to be able to move something, but exponentially better if when they move and touch something, there's a sensation that they touch something, and we're fortunate to be working on that too. Okay,
well then quickly I want to get back to Mike McLaughlin. You're already there with his arm. I mean, there is sensory input. I mean, we've been talking about output. There is sensory input, is that correct, with
That's right.
Temperature, pressure,
vibration. So you can run your fingers across, and you can sense, for example, surface texture. We've only just begun to really explore the use of those sensors. So we have something on the order of 107 sensors in the arm. So we can sense joint positions, we can sense touch, and we've only just begun to explore how to use this. I always say we're probably using 1 or 2% of the capability of the arm right now. But we have demonstrated in amputees the ability to stimulate the residual nerves that were left after the amputation, so that the patient can close their eyes and it can tell you which finger they're touching on the prosthetic limb. And so it's really quite remarkable, and they will tell you that they're sensing as if you're touching their native finger.
That's amazing. Okay, I want to leave time for just a couple of questions, but before we do, the very last question for the panel, Andy, since this started with you, I didn't even mention the animal studies that you've done. They're remarkable. He has trained monkeys in a very similar way to feed themselves by thinking, using a prosthetic arm. This is a major leap ahead because you've moved into a human trial or human experiment. Did that alone push the science? How did it help taking that step, and where do you see it going?
Well, if I could just back up a little bit, a couple of questions you talked about what this research is like, and I compare it to like an athletic competition. We had this 28 days where it was really intense and everybody was working hard, but if you look back for the whole thing, I look back at it and we've been working hard the whole time. It's been a big team, and it's been a heck of a lot of work, a lot of hard work by a lot of people. And so we started with the animal studies, which are in a way difficult because it's hard to train animals to do some of this stuff. They just don't get the concept of using their hand to operate tools. So that's a lot harder. It's a lot easier with humans to do that because you can just explain to them what you want them to do. But what we've done with the animal studies is we're recording from individual neurons, and that gives us a richer set of information to draw from, and we can translate that into much more elaborate control of the arm. And as you said, we're moving into human studies in that as well, and we're seeing this very elaborate kind of movement control, so you get really natural control. We're actually making use of this rich arm that we have now and controlling that in a much more natural way.
Yeah, and that's work that they have not published yet. It's very exciting. It takes off from this work, and also, according to Mike Boninger, they're still interested in working with the ECOG array because it's more practical. It's not as invasive, and it may, for various other reasons that we don't have time to get into, it may actually work better in a number of ways. Maybe not as precise, but maybe better and more practical for clinical use. Now, I know we have to wrap up, but does anyone have questions? Joe.
I'm sort of curious whether the brain signals are unique from individual to individual. In other words, if Derek Jeter and I are both trying to hit a fastball, he's amazing enough. Is it basically the same? Does it look the same in our brains at least?
So the basic principles, the language is similar. It's the same, actually. So we understood the language even in monkeys, and then we were able to apply that directly, the same language, to the humans. So the language is the same. Obviously, the details, which we aren't really capable of seeing yet in the resolution that we need to be able to tell the difference between you and Jeter. But
we're confident they exist, the differences.
Anybody else? Jim.
Yeah, I have a question for Tim. It sounds like it's such hard work going through the training and stuff. And I was wondering, did it feel, when you were working on moving the person and moving the arm, did it wear you out, like physical activity? Did you sense being tired at the end of the sessions?
Doing this, knowing what we were doing, obviously adrenaline was pumping through me the whole entire time. So no, actually, during the study, as we were working, I felt great. You know, I told Wei many times. He's like, okay, we got 15 more minutes, and I'm like, let's add, let's do 20 minutes, let's do 25 minutes, let's keep going. On the way back home, you get back in the car and then going home, that's when it hit me, and because it is very mentally exhausting, trying to move something that you can't move for hours, for 6 days a week, for 4 to 5 hours a day. It definitely gets to you. So there definitely was some mental exhaustion there. But during the time, I was pretty psyched up to just keep going at it, yeah.
Great. Josh?
Very briefly, with the sensors, are you doing it from the flexible electronics or the electronics that remain implantable permanently?
So yeah, I think that is the direction that people are very actively working on. So there's other type of materials that you can potentially use, and even there's publication about using like silicon, different kind of materials to get more flexible medium for that. And one of the emphasis here we do is that in terms of doing clinical and translational research is that we want to be able to use the clinically approvable or clinically proven technology to sort of developing the system early on, and that's why some of the technology we're right now using more sort of mature ones, but yes, there are definitely those kind of developments.
How about one more here?
Does the difficulty go up exponentially when you get to the point of using 2 fingers to pick up a coin as opposed to just using the whole hand to pick up a container of milk? Or once you master the technology, is it the same technology though?
So yeah, so we start obviously with the easier thing. So moving a whole hand is easier than moving individual fingers. And the whole thrust of the research project is to get to higher degrees of control, which would be more fingers, more incremented movement, more individuated movement. And that is definitely more difficult, not only from an engineering point of view, but for the subject to learn. But we are working very rapidly toward that. That's our main goal.
So I think one of the things that we're also looking at is, so we're working across multiple communities. So we're also working, for example, in telerobotics, where I want to be able to operate the robotic system at a distance. And particularly if you're at a long distance, say you're in outer space or something, you're trying to control a robotic arm, it requires that you have a lot of local autonomy. And so if I'm holding something and it begins to slip, I very quickly will grasp it, and that's a reflex action. Okay, it doesn't go all the way up and all the way back down. And we're beginning to look at these types of things too, to provide basically kind of local automated processing capability, so that things that require high dexterity, the arm can actually help, based on sensory feedback, decide whether I'm grasping it tight enough, or is it starting to give, those types of things that help take some of the cognitive burden, potentially take some of the cognitive burden off the individual operating the arm.
Well, I think we should probably wrap up. We've got 2 more panels, and I encourage you all to join those. I think these guys would welcome private questions. I would hope so, because I know that there's a lot more I want to ask them. So I would like to thank them for being here. I'm fascinated by this work, and I look forward to seeing what you all do in the future. Thank you for being here.
Yeah, thank you.